Hello, this is Professor White, and today I'll be discussing about the structure and function of macromolecules. Now, what are macromolecules? Now, macromolecules are mostly polymers. So when you say polymers, these are large molecules consisting of many identical or similar building blocks linked by bonds. Now, monomers, these are subunits that serves as the building block for polymers. Now, there are a limitless variety of polymers that can be built from small sets of monomers. Now, this is usually inherent in sibling differences, which results from variations of these polymers. Normally, there are about 40 to 50 common monomers that form a macromolecule and others that occur rarely. Now, small molecules that are common to all organisms are ordered into unique macromolecules. Now, how do cells use organic compounds? Now, biological organisms use the same kind of building blocks. Now, all macromolecules, large complex molecules, as we have to say, have a specific function in cells. Now, other than water, macromolecules make up the large percentage of mass of a cell. And in order for these macromolecules to be tied up together or linked into chain, there are two processes that's involved in the formation of macromolecules. First is the condensation reaction. So this is also called dehydration reaction. This is when two molecules become covalently bonded to each other through the loss of a small molecule, and usually it's water. Now, the second one is your hydrolysis, meaning there's a separation of two molecules by addition of water. So when you say condensation reaction, it's a dehydration, remove the water, and then your hydrolysis is when you try to separate molecules and, of course, add water. Now, large polymer forms from smaller monomers. Now, new properties emerge when this happens. Now, the living cells require or synthesize the following macromolecules. You have your carbohydrates, lipids, proteins, and nucleic acids. Now, the first macromolecule we'll be discussing is your carbohydrate. Carbohydrates are used as fuel building material. Now, carbs are sugars. Now, there are main types of carbohydrates. You have your monosaccharides, disaccharides, and polysaccharides. As you can see here in the diagram, monosaccharides are simple sugar. Disaccharides, that means there's two sugars covalently attached, and a polysaccharides, many sugars are covalently attached. Now, let's discuss about monosaccharides. Now, monosaccharides generally have a molecular formula CH2O. Now, glucose is an example of a monosaccharide, which is C6H12O6, which is the most common. And in aqueous solution, water solution, they may form rings. And this is actually a major nutrient for the cell. So that is your monosaccharide. Now, disaccharides, these are two monosaccharides joined by glycosidic linkages. Now, glycosidic linkages is a covalent bond formed between monosaccharides. And sucrose, or your table sugar, is the most common example of a disaccharide. So in this particular diagram, you'll see how two glucose are attached together to form your maltose, sucrose, and then there's a condensation happening to form your disaccharide maltose. Now, the next kind of carbohydrate is your polysaccharide. Now, 100s to 1000s of monosaccharides long comprises your polysaccharides. Starch is a very common example of polysaccharides, which is a storage of plants. Glycogen is another form of polysaccharides, and this is usually the storage for animals. Now, cellulose is another example of polysaccharides, which is a major component of the tough plant cell wall. And last but not least, the chitin. Chitin is actually a polysaccharide that is usually found in the exoskeleton or outside coverings of your arthropods or insects. Now, let's take a look at your starch and cellulose. As you can see here, they form rings in aqueous solutions. And the letter A is your alpha and beta glucose ring structure. The second one, which is letter B, is the starch. And the third one is your cellulose. Now let's go to lipids. Now, lipids are mostly hydrophobic. We know that hydrophobic are water-fearing, okay? And there's little or no affinity to water. So lipids are not really fond of water. Now, lipids are used for energy storage and structure. And there are many types of it: fats, phospholipids, and steroids. And we'll take a look at that in a few. We'll start first with fats. Now, fats are large molecules, but not polymers. Now, fatty acid is a long carbon skeleton with carboxyl group head and hydrocarbon tail. So you can see here on this example. So the whole long chain right there, your hydrocarbon tail, and then the top part is a carboxyl group. So that is your fat. So here's some examples: stearic acid, oleic acid, linoleic acid. The next one is your triglycerol or triglyceride. These are 3 fatty acids linked to one glycerol molecule. So as you can see here, 3 fatty acids and water. So you can see over here, so that's your triglyceride. Now, there are 2 types of fats: saturated and unsaturated fats. So when you say saturated fatty acids, now these are fatty acids containing no double bonds between the carbon atoms composing the tail. And normally, saturated fatty acids are solid at room temperature. However, your unsaturated fatty acids has 1 or more double bonded carbons in the tail. As you can see, the difference between the two, the one has what you call a kink. So that's the difference between saturated and unsaturated fatty acids. And unsaturated fatty acids are normally liquid at room temperature. Phospholipids. So there's another type of lipids, is phospholipids. Now, phospholipids has 2 fatty acid tail linked to one glycerol molecules. They have ambivalent behavior towards water. When in contact with water, they form a micelle or cluster. And normally there's a side of the phospholipids that are water-loving and hydrophobic too, which is your water-fearing side of your phospholipids. And when we talk about the cells in the next session, you would be able to see phospholipids in the cell. Another example of lipids is your steroids. Steroids are lipids characterized by a carbon skeleton consisting of 4 interconnected rings. I'm sure you guys have heard steroids in athletes or people working out, but these are because steroids are like hormones. Okay, so another is your cholesterol. Cholesterol is actually an important steroid that is a common component of the animal cell membrane. Now, there are many hormones that are steroids produced from cholesterol. Here are some examples: testosterone, cortisone, vitamin D, and this is basically letter D is your cholesterol. Now, the next macromolecules we're going to cover is your proteins. Now, the molecular tools for most cellular functions is, of course, tied up with proteins. Now, this is used for structural support, storage, transport of other substances, signaling from one part of the organism to another, movement, defense against foreign substance or immunity. And also, your proteins have what we call a 3D shape of conformation that fits whatever function it ought to do. Now, proteins are polypeptides. Now, polymers of amino acid connected in a specific sequence are polypeptides. Now, amino acids, these are organic molecules possessing both carboxyl and amino group. As you can see here in the structure, this is basically the structural form of our protein. And the acidity is determined by the side chain. So it's not really important for you to be technical, but I want you to recognize that this is the general structure of your protein. Now, peptide bonds are formed when an enzyme joins amino acids by means of condensation. As you can see here, the polypeptide chain forms linking these amino acid by a peptide bond. Now, as I mentioned earlier, proteins has a certain conformation or shape, which determines the function or the role of the protein and is a result of a linear sequence of amino acid in a polypeptide. Now, there's folding, coiling, and the interaction of multiple polypeptide chain creates a functional protein. Now, there are 4 levels of conformation: primary, secondary, tertiary, and quaternary. So let's take a look at the primary structure. So primary structure, usually they are unique linear sequence of amino acids. So it's just like straight chain. A change in one amino acid can actually affect every other level. For example, point mutation in hemoglobin, just like in sickle cell anemia. The second structure is your secondary structure. Now, there's a hydrogen bonding going on between amino and carbonyl group of amino acids, and secondary structures formed can be alpha helix and beta sheets. Now, alpha helix, usually this is common in fibrous proteins that creates elastic property. And then for the beta sheets, these are anti-parallel chains of, it's kind of like pleated. So that's the difference between the alpha helix and your beta sheets. The next one is your tertiary structure. Now, tertiary structure have irregular contortions from bonding between side chains of various amino acids. And last but not least, we have your quaternary structure. This is the overall protein structure that results from aggregation of tertiary units. So this is basically the overview of all those structures: primary structure, secondary, tertiary, to quaternary structure. Now, what is denaturation? Denaturation is basically the unraveling and loss of native conformation of the protein. Sometimes this can be due to heat. When proteins are subject to heat, they denature, pH, salts, and etc. Some can renature exactly, but others cannot. An example of denaturation that's happening is when you cook an egg, the proteins in that egg whites. As soon as it turns from clear to white, then denaturation happens. And last but not the least of our macromolecules is your nucleic acid. Now, nucleic acid store and transmit hereditary information. As we know, that gene is a unit of inheritance. Now, nucleic acids, we have your DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA is ribonucleic acid. Now, DNA is like a computer software. Proteins are like hardware. Now, genetic info flows from DNA to RNA to protein. Let's take a look at the DNA structure. DNA structure has a polymer with an information-rich sequence of nucleotides. And there's parts of your DNA structure. We'll start first with the pyrimidine. Now, pyrimidine is a 6-membered ring made of carbon and nitrogen atoms. And there's 2 types of pyrimidine. You have your cytosine and thymine. The next one is your purine. Now, purine is a 6-membered ring fused to a 5-membered ring. And you have 2 purines. You have your adenine and guanine. Now, phosphodiester, these are covalent bonds holding the nucleotides together. Now, DNA is a double helix, meaning there are 2 chains of nucleotides that spirals around an imaginary axis. Now, the bond that holds these chains together is hydrogen bonds. Now, the adenine usually pairs with thymine and cytosine pairs with guanine. And the 2 strands of DNA double helix are complementary. Now, RNA, if DNA is double-stranded, RNA is just a single-stranded. Now, there are 4 kinds of nucleotide monomers. You have your adenine, uracil, cytosine, and guanine. As you can see, there's no thymine in RNA, but the U, which is right over there, is uracil. Now, the key players in protein building processes is your RNA. And there are 3 of those: the mRNA, or messenger RNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA. Now, why is DNA and protein important? It is important because inheritance is based on precise replication of DNA. We can use the DNA and protein as tape measures of evolution. Also, the linear sequence of nucleotide in DNA molecules are passed from parents to offspring, and more distantly related species have chains that are less similar, and DNA has that information which we can be provided with. And I hope you learned something, and I'll see you in class. Bye.